Now we're going to go ahead and start the living kingdoms. Remember, viruses did not have all the characteristics of life, therefore they were not considered living. Bacteria, however, are living. They do have all the characteristics of life. Bacteria are also considered prokaryotic living things. They have DNA but no membrane-bound organelles. Bacteria are the only living things that are considered prokaryotes, not having a nucleus. Now, we did divide them up into two domains, archaea and eubacteria. Now, under archaea, you have kingdom Archaeobacteria, and then under eubacteria, you have basic bacteria or eubacteria. Now, the Archaeobacteria, we said, live in harsh environments. According to evolutionists, this would be the more ancient bacteria. Now, bacteria previously, about 50, 70 years ago, maybe more, were placed in one kingdom called Kingdom Monera. Some resources, if you look online, in some textbooks, it will say Kingdom Monera. Now, bacteria are unicellular, meaning they only consist of one cell. So if they come together, if many bacteria come together in a pile, it will never form a tissue. Remember, cells make tissues, but not with prokaryotes, not with unicellular organisms such as bacteria or protists. A bacteria, if they congregate, it's going to be called a colony. Now the size, they're about 1 micrometer. This is, even the smallest bacteria is larger than the largest virus. Bacteria will reproduce mainly through an asexual process of 1 to 2, and that's called binary fission. Now, bacteria can reproduce in asexual means, and that's actually by conjugation. What conjugation is, a bacteria will make a little bridge or a little pili tube, and this little pili will connect itself with another bacteria. And it will break off, it will shed a little bit of its DNA and pass it through the pili tube into the other bacteria. That other bacteria could merge the DNA with itself. A bacteria can do this to a dead bacteria or a non-functional bacteria and bring it back to life. Now, bacteria do have positives. There's positive bacteria in your body. I know it's a little gross, but when you go to the bathroom and maybe you have different color poop or feces, that's because of the bacteria. If you scrape the back of your tongue, that's why it's so important to brush your teeth and brush your tongue. If you scrape the back of your tongue and it smells, it'll smell like poop. That's because of bacteria. Now, there's helpful bacteria in your intestines, and there's also helpful bacteria that breaks down waste in nature, breaks down dead animals. Bacteria can also be used for making yogurt and tanning leather. So it can be used to make food as well as using it for different processes. Now, bacteria can also be very, very harmful, such as food poisoning, such as botulism. And there's a lot of deadly bacteria. Unlike viruses, in which viruses have hundreds of different shapes, bacteria have 3, 4, 5 basic shapes. We're going to focus on the 3 basic shapes, which is coccus, bacillus, and spirillum. Coccus is circular or spherical. Bacillus is rod-shaped or more rectangular. And spirillum is spiral or corkscrew shape. Now there's another one called vibros, and vibros is more of a comma shape. Here's an electron microscope of coccus bacteria. Electron microscopes deal with shades of black and white and gray. Here's bacillus. Salmonella is an example, and spirillum. Okay, so we talked about the 3 basic shapes that you can see at the top, but now there's arrangements. If they're in a group of 2, you're going to add the prefix diplo. Think of di, 2, di and bi. So diplo meaning 2. So you can have diplococcus, you can have diplobacillus, or you can have diplospirillum. So this is 2 of the certain shape of bacteria. So if you have 2 bacillus, 2 little rectangles together, it's diplobacillus. Then you have streptococcus. Now, streptococcus is commonly known as strep throat. Streptococcus is when you have a chain or a line that's strepto of coccus bacteria. So you can have streptobacillus, which is a line or chain of bacillus bacteria. And then the last one, staphylo. If you ever heard of a staph infection, staphylo means cluster, if you have a cluster of bacteria. So you can have a cluster of spirillum, so staphylospirillum. You can have staphylococcus. Now we're going to move into memorizing and knowing the structures of the different living organisms. We're going to start with bacteria structures, and I would encourage you to put these on index cards or Quizlet. The first one is the cell wall. This is a covering over the cell of bacteria. Scientists have actually used different stains that they add to bacteria to see if the bacteria has a protective cell wall or a double cell wall. Now the stain is a purple color, so if the bacteria stays purple, it's going to be gram positive. This means that it can be killed by penicillin, killed by antibiotics. Now remember, penicillin is an antibiotic that can kill bacteria. It's a medicine that can kill bacteria. Penicillin cannot kill viruses. Now, if they add this stain to a bacteria that is gram negative, it will have a 2-layer cell wall, which is harder to kill, and it does not keep the pink stain. So if it does not retain the stain, it's gram negative. And that's, we have some pretty bad bacteria here that could be very harmful. It can't be killed by antibiotics. A capsule. A capsule surrounds the cell wall, so bacteria have 2 cell coverings. A cell wall first, and then a capsule. Then it has a plasmid. When we learned about the lytic cycle and lysogenic cycle of the viruses, I showed you that there was a circular DNA inside bacteria. The plasmid is the circular DNA of the bacteria. So remember, the bacteria does have DNA but no nucleus. And it will usually become circular when it's ready to go through binary fission. Now when we have the pili, remember the pili can be used as a bridge. It's a small hair-like appendage. An appendage is like an arm or a leg, but it's a small hair-like appendage that acts as a bridge. It connects to another bacteria to transmit its DNA. And this happens in the sexual process of conjugation. Now, bacteria can have some forms of movement. They can have these long propeller-like, whip-like structures. They look like little hairs, but these are different from pili. They're not a bridge, but these are like little whips. It allows it to move. Now the flagella, it can have 1 or 2 or 3 or 4. There's not a lot of them, but probably at the most, 4 or 5, 6. Usually 1 or 2. We already learned what the cytoplasm was, cell fluid, and then we learned what ribosomes were. Okay, it's a site of protein construction. So bacteria will not have membrane-bound organelles, but it will have DNA as well as ribosomes and cytoplasm. Just a reminder before I move on, remember the capsule is the covering of the cell wall of bacteria. Do not confuse this with capsid. Capsid was the covering of the virus, while the capsule is the covering of the bacteria. Here you can see the capsule as well as the cell wall and plasma membrane. You can see the cytoplasm, the ribosomes, okay, the DNA, and then you can see a little pili being made, as well as the flagella for movement. Make sure you know the 2 types of reproduction, asexually for binary fission, happening very quickly, around 20 minutes, and that bacteria divides equally 1 to 2, and then you have conjugation, the formation of the pili to transfer DNA or a piece of the DNA to another bacteria, so that's considered sharing, so that would be sexual reproduction. So if I break off a piece of my brain and I hand it to another person and they put it inside theirs and they give me a piece of their brain, okay, that would be considered sexual reproduction because it's a sharing of that information.